Okay, this is the ova and this is fusion, and now after fusion the cell start dividing, and this phase of division, the as soon as the cell start divide, this is known as cleavage. Clear? The cell will transform into various stages like one morula. Okay, so as a result of cleavagic activity, the cell divides, and these cells which does not increase in size but increase in number are known as blastomeres. Okay, bache? So they keep continuing divide, and they reach to up to 8 to 16 cell stage. That is more important. Okay, what is the cell stage? 8 to 16. And this look like a berry I have not eaten at all, and even you might have eaten it, a berry, mulberry. Okay, that mulberry like structure. Okay, it's a small tiniest structure. That tiniest structure consisting of 8 to 16 cell after division of zygote is known as morula. Okay, these all cell which are clustered or protruding outside are sticky in nature. Okay, so they roll down from the isthmus. Okay, after 6 to 7 days of um, fertilization. Okay, and they will become the second one known as blastocyst. Okay, so second thing, the second stage is blastula stage. Okay, this is the thing which get implanted into the endometrium. Now listen. Okay, so what will happen? You can see here. This is morula. Okay. Now, when the blastocyst reaches the uh, uterus, okay, and try to embed into the uterine wall. Okay, what happens there? There is a secretion of some nutrients into this cell, and this nutrient is known as uterine fluid. Okay, and the cell arrange accordingly. Now, how the cell arrange in the blastula? Look here. Some of the cell make a layer, and that outside layer is known as trophoblast. Okay, and also known as trophoectoderm. And there are a cluster of cell here. Okay, by the secretion of uterine fluid, and also the trophoblast secretes some nutrients, these cluster of cell arrange at one portion. Look here, they are arranged at one portion and they form a knob. This knob is known as embryonal knob. This cell which are in contact, listen, these cells which are contact of the embryonal knob are known as Rauber's cell. Now listen, this embryonal knob is usually or is helpful in the formation of germ layer. Okay, the embryonal knob participate in the formation of germ layer, and the trophoectoderm participate in the formation of extra embryonic membrane. Clear? So you can see, in case of blastocyst, the blastula, there are two kind of cell. One, the outer cell known as trophoblast, or the trophoectoderm, and the inner cluster of cell arranged at a corner or at a point known as embryon knob. Okay. Because of these arrangement of secretion of trophoblast and uterus, there becomes a cavity known as blastocele. This cavity which has become inside this blastocyst, this cavity is known as blastocele, and the whole structure is blastula. Okay, the cell which are in close contact with the embryonal knob are known as Rauber cell. These cells degenerate later during the placenta formation. Okay, and those cells on the outer surface are the trophoblast cell, which participate in the formation of extra embryonic membrane like chorion, amnion, yolk sac, allantois. So these are formed by the uh, trophoblast, and the germ layer, mesoderm, ectoderm, endoderm will be formed by the embryonal knob. So what exactly is implantation? Now we have a structure known as blastocyst. The blastocyst get buries inside the endometrium, and the endometrium, what it does, it's cover up the blastocyst completely like this, है ना? Cover up. So this type of implantation is seen in humans. After the implantation, this endometrium which have covered up, or the portion of endometrium that has taken implantation, is known as decidua. Okay, what exactly is a gastrulation? What is gastrulation? देखो, gastrulation is very important process. Okay, because any embryo will form, it will require germ layer. Okay, because we have different types of organ forms of different germ layer. Or what happens in the gastrulation, a critical changes occurs in the embryo, and in this, three primary germ layers are formed. Okay, you know that the blastula was single walled. It was having only trophoblast. It was having only trophoblast. And now this blastula will change into double walled gastrula. Now listen, what will happen in this gastrulation? Okay, these are the trophoblast cell. Clear? As soon as implantation occur, there also occur the formation of chorionic villi that participate in the formation of placenta. So these are the trophoectoderm, this is the chorionic villi, and the inner cell of mass is known as embryonal knob. This is the blastula. Okay, now the some changes will occur in the blastula and transform it into gastrula. This is chorionic villi, and this is trophoectoderm or trophoblast, and this one is embryonal knob. So what happened in this case? The cell of embryonal knob arranged in a disc-like pattern. Achha, what are these? What are these upper one? Rubber cells, cells of rubber. They will degenerate now. They will degenerate, and degeneration of this, remember, degeneration of this cell will result in formation of a cavity known as amniotic cavity. So what is happening here? These are the what cell? Trophoblast cell, and the rubber cells have now been degenerated. Okay, so what exactly happened to the embryonal knob? Embryonal knob have arranged into the disc-like structure like this. Okay, they were scattered before, and now they have arranged into a disc-like structure. They have settled on one point. Okay, so Some of these embryonal knob cells will be of columnar shaped cell and some will be of cuboidal. This is columnar, cuboidal. The cell which are arranged above these cells are known as epiblast. The cells which are arranged below are known as hypoblast. So what will happen here after the degeneration of Rauber cell? There become a space. Okay, so these columnar cell get shifted towards the position of Rauber cell. So now they have come to the position of what? Rauber cell. During this, what happened? The cuboidal cell remain behind, and this lead to the formation of two layer now. One above and second is down. Process occur here is delamination. Okay, now what will happen? These cells which have detached or delaminated from the embryonal knob continues to multiply, and this multiplication result in a formation of first layer known as endoderm, and this endoderm will give rise to primitive gut. So we have understood the formation of endoderm. So two more layers are left. This two layer structure is basically the gastrula. Gastrula has been started formation. The gastrula formation is started. Okay, now two more layer are left. So let me number these diagram. Okay, 1, 2, 3, 4, and the next will be 5. Okay, so what happens here in the formation of mesoderm? This end of the epiblast, cephalic end. Which end? This end of epiblast cell is known as caudal end. So some of the caudal end cell, they detach and multiply and give rise to the middle layer known as mesoderm. Now the last is, so what will happen in the ectoderm formation? This is the trophoblast cell, and these are epiblasts. Now I'm not drawing the inner two one, I'm just drawing the outer one, that is ectoderm. This epiblast cell continues to multiply without being detached, without detaching itself. So there will be a formation of a layer ectoderm. Clear? And inside this layer, you will see this is endoderm, and the scattered pouches are known as mesoderm. This is how the three germ layers are formed. Now, extra embryonic membrane. That main role of chorionic embryo or extra embryonic membrane is to provide nutrition for the respiration and for the excretion of the embryo. So these extra membrane. So now the trophoblast will come into play. This trophoblast will uh, participate in the formation of extra embryonic membrane. So there are 4 kinds of extra embryonic membrane. Number 1 is amnion, number 2 chorion, number 3 yolk sac, and number 4 allantoid. This is what embryo. Okay, we know that this is the embryo which is forming here, and there are some layer like these are the extra covering of the embryo. So that is why they are known as extra embryonic membrane. The in embryo is a sac-like structure, yolk sac. What we call it? Yolk sac. And allantois. Okay. This is covered by a amnion membrane, and the whole structure is covered by chorion membrane. All of these are composed of trophoblast. So there will be a cavity between embryo and amnion layer. This cavity is known as amniotic cavity. Consists of a fluid known as amniotic fluid, and this fluid is very helpful in determining the sex of the child and also some genetic disorder, and it also protect the developing embryo from the mechanical shock. So suppose if there is a fluid here, so you can see that if is there any force applied here, so this fluid can prevent the direct contact of the force, है ना? So that is how this amniotic fluid is helpful in uh, providing protection from the mechanical shock. So what are these? Are arising from the fetus and also connecting with the maternal blood. These are nothing but the maternal blood vessel, and these maternal blood vessel will be in a tissue known as placenta, wrap in a cord-like structure known as umbilical cord. So these are the extra embryonic membrane. So I think we are clear here.